Now, here's not just an amazing story, but a spectacular one. And I mean Ouroboros style. In order to create, you must destroy. DNA damage and inflammation key to memory formation. I mean, that's amazing. Hello and welcome back once again to another episode of Focus, and happy Easter. DNA damage and inflammation key to memory formation. Researchers unveiled a groundbreaking discovery that DNA damage and brain inflammation are a vital process for forming long-term memories, particularly within the brain's hippocampus. Contrary to previous beliefs, associating inflammation with neurological diseases, this study highlights inflammation's critical role in memory formation through the activation of the Toll-like receptor 9, or the TLR9 pathway, following DNA damage in the hippocampal neurons. These findings not only challenge conventional views on brain inflammation, but also caution against indiscriminate inhibition of the TLR9 pathway, given its importance in memory encoding and the potential risks of genomic instability. Key facts. 1. Unexpected memory mechanism. Brain inflammation and DNA damage in the hippocampal neurons are essential for the formation of long-term memories, facilitated by the TLR9 inflammatory pathway. 2. DNA damage and repair. The study found that the hippocampal neurons undergo cycles of DNA damage and repair, organizing them into memory assemblies, crucial for storing episodic memories. 3. Caution against inhibition. While drugs targeting the TLR9 pathway may offer therapeutic potential, their use requires caution due to the pathway's essential role in memory formation and the risk of genomic instability. And the source is from the Albert Einstein College of Medicine. Just as you can't make an omelette without breaking eggs, scientists at Albert Einstein College of Medicine have found that you can't make long-term memories without DNA damage and brain inflammation. The surprising findings were published online today in the Journal of Nature. Inflammation of brain neurons is usually considered to be a bad thing, since it can lead to neurological problems such as Alzheimer's and Parkinson's disease. Apologies. Said study leader Elena Radulovic, MD, PhD professor in the Dominic P. Purpura Department of Neuroscience, professor of Psychiatry and Behavioral Sciences, and the Sylvia and Robert S. Olnick Chair in Neurosciences at Einstein. But our findings suggest that inflammation in certain neurons in the brain's hippocampal region is essential for making long-lasting memories. The hippocampus has long been known as the brain's memory center. Dr. Radulovic and her colleagues found that stimulus sets off a cycle of DNA damage and repair within certain hippocampal neurons that leads to stable memory assemblies, clusters of brain cells that represent our past experiences. Elizabeth Wood, a PhD student, and Anna Sivarik, a postdoctorate in the Radulovic lab, were the study's first authors at Einstein. From shocks to stable memories. The researchers discovered this memory-forming mechanism by giving mice brief mild shocks, sufficient to form a memory of the shock event or an episodic memory. They then analyzed neurons in the hippocampal region and found that genes participating in an important inflammatory signaling pathway had been activated. We observed strong activation of genes involved in the Toll-like receptor 9 or the TLR9 pathway, said Dr. Rodlovic, who is also director of the Psychiatry Research Institute at the Montefiore Einstein or the PRIM. This inflammatory pathway is best known for triggering immune responses by detecting small fragments of pathogen DNA. So at first we assume the TLR9 pathway was activated because the mice had an infection. But looking more closely, we found to our surprise that the TLR9 was activated only in clusters of hippocampal cells that showed DNA damage. Brain activity routinely induces small breaks in DNA that are repaired within minutes. But in this population of hippocampal neurons, the DNA damage appeared to be more substantial and sustained, triggering inflammation to make memories. Further analysis showed that DNA fragments, along with other molecules resulting from the DNA damage, were released from the nucleus, after which the neurons in the TLR9 inflammatory pathway was activated. This pathway in turn stimulated DNA repair complexes to form at an unusual location, the centrosomes. These organelles are present in the cytoplasm of most animal cells and are essential for coordinating cell division. But in neurons, which don't divide, the stimulated centrosomes participated in cycles of DNA repair that appeared to organize individual neurons into memory assemblies. Cell divisions and the immune response have been highly conserved in animal life over millions of years, enabling life to continue while providing protection from foreign pathogens. Dr. Rodlovic said, "It seems likely that over the course of evolution, hippocampal neurons have adapted this immune-based memory mechanism by combining the immune response's DNA sensing TLR9 pathway with a DNA repair centrosome function to form memories without progressing to cell division, which is otherwise known as cell aging, but resisting inputs of extraneous information. During the week required to complete the inflammatory process, the mouse memory encoding neurons were found to have changed in various ways, including becoming more resistant to new or similar environmental stimuli. This is noteworthy, said Dr. Rodlovic, because we're constantly flooded by information, and the neurons that encode memories need to preserve the information that they've already acquired and not to be distracted by new inputs. Importantly, the researchers found that blocking the TLR9 inflammatory pathway in the hippocampal neurons not only prevented mice from forming long-term memories, but also caused profound genomic instability, i.e., a high frequency of DNA damage in these neurons. Genomic instability is considered a hallmark of accelerated aging, as well as cancer and psychiatric neurodegenerative disorders such as Alzheimer's, Dr. Rodlovic said. Drugs that inhibit the TLR9 pathway have been proposed for relieving the symptoms of long COVID, but caution needs to be shown because fully inhibiting the TLR9 pathway may pose significant health risks. The study is titled Formation of Memory Assemblies Through the DNA Sensing TLR9 Pathway. and there's many other Einstein authors or co-authors. But like I said, to me this is an amazing story. I mean, the way it works, like I said, Ouroboros style. There can be nothing new without destruction. Thanks once again for tuning in to another episode of Focus. And like I said, happy Easter. Hey, thanks for sticking around. But you know what? I truly won't take up much of your time. It's Easter. So you know what? If you have them, go spend some time with your family. Tell them how much you love them. And have a great day. Take care. For links to the articles, check out the episode description. If you want to leave a comment, check out the trailer description for the email address. For topics like these and more, check out future episodes. Like I said, the world is an amazing place. There's a lot going on, and sometimes we just need to take a moment to focus.